Okay, coming in now. Yep.
Yep, all good. Well, thank you for that very nice introduction. That was probably more details than you needed to know about me, but I am obviously very interested in reproductive resilience. Thank you for inviting me to give this talk, and I'm really hoping that there'll be some people online and be some interesting discussion that comes out of it. So you might be wondering, what is reproductive resilience? And it's defined as the capacity of a population to maintain the reproductive success needed to result in long-term population stability despite disturbances such as climate change and fishing. We had published a paper on red snapper, but the one that really brought things together was this one in fish and fisheries with just an awesome co-author group. We were able to draw on expertise in stock assessment, modeling, genetics, dispersal, early life history, and reproduction to bring together this idea. And what it really focuses on is that spawn and recruit systems are complex. Marine fish have more diverse reproductive strategies than any other vertebrate. And these strategies are made up of fixed behavioral traits, as well as traits that vary with ecological context. And then you have two feedback loops: density dependence, which we talk a lot about in fisheries science, and fitness, which we talk less about, but I'm guessing with all the genomic advances we'll be talking about more and more. And then the other key point is that often in management, we think about year class strength. This plays a role in terms of fish abundance, obviously, and fishermen's observations of abundance. But in terms of reproductive resilience, this is just considered part of the natural system, and the really key time scale is that of the reproductive lifespan. So basically, when you mature and how long you live, how many years did you have to reproduce successfully? For very few fishes is that one. And for the fishes I'll be talking about, it's usually 30 plus. So why does this matter for fisheries assessment and management? It's difficult to manage species for which we do not understand drivers of future abundance, and this has been brought up a lot and is increasingly being brought up, including by stock assessment scientists. And of course, this is just going to get more difficult as ocean conditions change. And because we need a new recruitment conceptual model, what I mean by this is that the accepted dogma is really the sweepstakes recruitment success model that came out. Hedgecock was the one who published on this in the 1990s, and then he did another nice paper in 2011, and there was a lot of fisheries papers showing these really low effective to census population sizes, and coming to the conclusion that this meant there was sweepstakes reproductive success. A key problem is that this hypothesizes that reproductive success is largely random. And that would mean that fish don't evolve. And there's a whole lot of papers in science that suggest that that's not the case. And it's important for us to look under the hood and think about what that means. And again, obviously for reproductive resilience, the whole concept is that they have evolved systems to be successful in their environment. So what are they? And then I just want to highlight this paper that just came out last year that Mike Tringali led. It was really nice, a very nice educational experience for me to refresh my genetics. I'm not sure it was quite as nice for Mike. He spent a lot of time, he was very patient, making sure that I got all the concepts. But we used this pretty extraordinary data set we had on red drum. We've done genetic tag recapture and non-lethally sampled over 12,000 fish at a given spawning site, and then also had data on movement from electronic tracking and spawning aggregations from aerial surveys, as well as reproductive phase data. So we were able to look at that and evaluate whether red drum, in fact, do exhibit sweepstakes reproductive success. They, in the past, have been considered a poster child based on a really nice paper by Turner et al. But in fact, we did not find the low effective to census population ratio, excuse me, that you expect if you have SRS. We found about 0.4, and SRS is usually 0.001 or so. And this is increasingly being shown for other species as well. We use some published data on southern bluefin and silver sea bream to also look at lifetime reproductive success in all three of these species and compare the traits. And there's more and more coming out in the literature. that these very small effective population ratios were probably driven by sample size and not completely taking into consideration spatiotemporal effects. So many of the studies from the 1990s, 2000s, they would have genetic sample sizes of 200 to 300 over large spatial scales when you have metapopulations, and that causes problems. So it looks like I think we'll be seeing more and more fish being tested for SRS and showing that they in fact do not have that. So what's going on? We get these stock assessments that show stock recruit relationships that have shotgun data and don't seem to fit very well with the theory other than at 0, 0, which is not especially helpful, right? And again, a number of papers coming out showing that there's not a strong relationship between recruitment and adult abundance. My hypothesis is that's because we're taking terrestrial ecology for terrestrial vertebrates and applying it to marine fish, which have very different reproductive strategies. So in marine systems, you have both the fixed ratio strategy and the small egg strategy. Things like dolphin have the fixed ratio strategy, as do terrestrial vertebrates. In these species, their offspring are a fixed ratio of adult size, with offspring getting larger as adult gets larger. They have relatively low fecundity, even when you think about something that makes babies like rabbits. We're talking 84 in a year. And when they give birth, their babies stay put. So in marine fish, most of them have pelagic eggs. They're about 1 millimeter in diameter. You can have a size, adult to offspring size of 800 million to 1 in something like bluefin tuna. You have huge fecundities, and you have early life dispersal. And in many ways, their reproductive strategies are more like dandelions, who actually produce multiple batches that are dispersed with the wind, than they are like something like white-tailed deer. So I think it's really important that we begin to start thinking about bringing together these two groups that have been somewhat siloed, which is those studying reproduction and reproductive output, and those studying early life history and things that impact survival in those early life stages. So just to wrap up in terms of reproductive potential, it's really taking the life history traits and looking at them through this filter of reproductive success, highlighting the fact that movement reproductive strategies in marine fish are linked. Certainly for anything that has pelagic eggs, they're linked, and even for many species that have demersal eggs, they're linked because they undertake spawning migrations. Current conceptual models of life history and resilience don't capture this. We often think of R versus K, but small pelagic fish are highly R selected, and yet some of the most common stocks to collapse. So I think there's a lot in this space that we can do, and I'm really excited about the new tools that are coming online to do it. Traditionally, for reproductive potential, we look at SSB and total egg production. We acknowledge egg quality matters, sexual system matters, but we're just beginning to really integrate mating system, behavior, movement throughout the life cycle. So how did I organize my talk? I wanted to take this graphic that was in our paper and modify it a little bit, and then go through and use some of these decision criterion to talk about what sorts of things we're seeing, drawing on the experience we had leading the reproductive analysis for these 3 different stock assessments for red snapper, gag, and scamp. But before that, I'm going to start out talking just a little bit about the origin of traditional reproductive potential measures versus emerging understanding of warm water reproduction, because I think that plays a role as well. We need to start thinking about developing conceptual models that work for our species. So, I think anyone who's familiar with fisheries science, if you think of the classics, you're not going to leave out Hjort or Beverton and Holt or Cushing, all huge in fisheries science and developing conceptual models, all work in cold water systems. Then Tara Marshall, which you guys may not know, but she is a really good scientist out of the UK, and she was one of the first to publish on this idea that total egg production is a better measure of reproductive potential than SSB. She's actually in Scotland. So what does this mean? Thinking about cold water systems, right? They're often food limited. They have high seasonality and a short window in which offspring can survive. And because fish are ectotherms, this is going to impact their metabolism and thus the rate of gonadal development. So they usually have slow gonadal development, restricted spawning seasons, meaning short, and determinate fecundity. And this is a biggie, and I'll come back to this multiple times. But in cold water systems, you can sample a fish within the spawning season and count the number, if it hasn't started spawning yet, count the number of oocytes that are in secondary growth and estimate individual annual fecundity. And that's just not possible for most warm water fish. In addition, sequential hermaphrodism is really uncommon in these systems, at least sequential hermaphrodites that are fished anyway. So I worked with a number of folks that are reproductive experts in the South, Southeast US on this paper, this unified framework paper. And we drew a lot on what we've done with some of these assessments and across NIMFs and state and academia. And one of the things we did is we compared some key reproductive traits in Southeast US federally managed finfish, so there's 24 species, versus some common cold water fish. So in our species, that Southeast US federally managed fish, 100% have indeterminate fecundity. And what that means is that they're multiple batch spawners, and they recruit new oocytes during the spawning season. So you can estimate individual batch fecundity, that's just the number of eggs that they would release in any batch. But spawning frequency is based on proportion of spawners. So it's always at the population scale. You're not getting individual spawning frequency estimates. 96% of these species produce pelagic eggs. So movement and dispersal will be super important. Most have extended spawning seasons, and a third are protogynous. So I think that really highlights nicely when you think about that first graph and decision criterion that they're going to be a little different for warm water species than what we've typically seen in cold water. And I think another thing that typically is not included in stock assessments, you know, we include uncertainty, but not uncertainty in terms of reproduction, as far as I know anyway. And I'd like to highlight just how uncertain annual fecundity estimates are if you have indeterminate fecundity. So in these species, excuse me, fecundity, as I mentioned, is batch fecundity times spawning frequency. And spawning frequency is the number of times a fish spawns in a spawning season. This was originally developed for schooling small pelagics. Brilliant idea. Hunter and Masewitz really changed the game, and they were the first to realize that their fish actually had indeterminate fecundity. And as I mentioned, batch fecundity is estimated based on hydrated oocytes, and you can see that picture down in the middle. Those clear round blobs are hydrated oocytes. It's pretty straightforward. You can do the gravimetric method with a weighed subsample, count your hydrated eggs, and extrapolate to the ovary. Batch fecundity is more, spawning frequency is more complicated. It uses spawning fraction, which is the percent of females with hydrated oocytes or day one post-ovulatory follicles. Those are POFs. They're just a spawning marker that's left after the egg is spawned. So you can tell that fish recently spawned. They usually last about 48 hours. You can use both of these markers, but then you need a correction factor to standardize to 24 hours, 1 day, so you can get your accurate spawning fraction. And why that has to be 24 hours is because it's the reciprocal of that fraction that gives you the number of days in the spawning season. Well, it gives you your spawning interval. I'm sorry, what I have written there is a little incorrect. So it gives you your spawning interval. So say you had 1/5, right? So you're gonna have 20%. So it would be 1 spawn every 5 days. That would be your spawning interval. And then you take the number of days in your spawning season, divide by 5, and that would give you your spawning frequency of the number of times the fish is spawning in a season. But the assumption is that movement is not affecting this spawning fraction indicator. I'm going to use the work we did on red snapper from CDR 74 as an example. They're gonochoristic and typically, measured with either, well, total egg production usually. So I want to talk a little bit about some of the issues with that. So certainly red snapper are one of the more data-rich species. Hundreds of thousands of records that were actually had an assigned sex. More than 10,000 females with reproductive phase. That's just the gonadal development that tells you where they are in the spawning season. More than 1,000 fish had batch fecundity. So when I first looked at these numbers, I was like, wow, this is just incredible. We never see numbers like that. So, but then, of course, there was the stock ID or population structure workshop, and based really on fishing pressure, they wanted to break the stock into 3 spatial regions. For reproductive data, we really had data for East and West using Mississippi as the break point there. And then the data comes from 3 periods of very different stock status: a highly overfished period, rapidly recovering, and stabilizing. And initial data visualization suggested that we were seeing different reproductive effort with those 3 different times. So we wanted to look at that broken out by time period. And then again, when you look at batch fecundities, I was so excited that we had more than 1,000 fish. But if you look at those 2 graphs to the right, you see a whole lot of white space after about age 14 or so. And these are fish that live to 57. So not a lot of fish in the older ages, only 23 that were older than 15. So again, using this as an example, the spawning frequency for red snapper in the Gulf, and this is just combining the groups right now, range from 39, the regions, range from 39 to 109 spawns per year, depending on spawning marker used and the definition of the spawning season. We have not in the past had a standardized method to estimate spawning season. Obviously, given that the number of days in your spawning season greatly impacts your spawning frequency, that's really important. And these differences in spawns per year result in a mean annual fecundity ranging from 10 to 29 million eggs. That's a pretty big range, right? Threefold. So one of the things we did as our part of the workshop and working with some great people was develop a method to quantify the core spawning season. And I should give a shout out to Claudia Fries. So she has been just instrumental in all of these reproductive analyses for stock assessments, and she helped develop this method. But even with this standardized method, using logistic regression to look at 50% spawning when fish are developing and starting their spawning season and when they're finishing their spawning season, you still get quite a range. Now you're from threefold to twofold, just based on what spawning marker you use and your spawning fraction. These aren't too bad, 18% to 20%, that's pretty similar. We usually do not get similar percent hydrated and percent POF, and you should if there's not a problem with movement. And we do a lot with movement ecology. Obviously, it's part of our lab's name. So what we find is that movement affects spawning fraction more often than not. And in fact, all the species we've studied so far it has. And in fact, we had the study on spotted sea trout and had a site that fish were moving to just to spawn, so 97% spawning fraction. And we had, they make courtship sound. We had underwater recorders so we could show that the courtship sound was daily, spawning was daily. But then we used telemetry to track the fish moving to and from those spawning sites. We were able to show that although spawning was daily at the population scale, it wasn't at the individual scale. And in fact, using those movement-based spawning frequencies, female spotted sea trout were actually spawning about half as frequently as what had been traditionally reported. So thinking about BOFs, right? So BOFs, we've been discussing BOFs for a while, and they were first developed looking at redfish on the West Coast, and then highlighted with some of the work in the cold water systems and a European cost initiative looking at reproduction. And then a different Marshall, not Kara Marshall, published that paper in Science about how fecundity increases faster with body weight, faster than body weight, and that you are not getting the right measure if you use body weight. He was looking at just batch fecundity, by the way, so it didn't even take into consideration spawning frequency. So you would expect in red snapper, they live to 57. You'd expect older fish to disproportionately contribute to reproductive success. That's partly how you end up having a long-lived life history strategy. And we know that fecundity increases with older red snapper. But what happens if you've killed most of the BOFs? And in that severely overfished period, we were often seeing, maximum ages that you would get a decent sample size of about 8 or 9, right? So you can see over on the right-hand side that oval that's just showing BOF space. And you can see for batch fecundity, right, there's not much there. And then that's spawning fraction on the bottom. And why it jumps up and down like that is because when you have very few fish, you often get either 0% or 100% spawning fraction. So if you've severely overfished a stock, and BOFs really are important, and you've killed most of them, the stock typically crashes or it compensates. And I would make the case that red snapper have compensated, and I have a little more on that, but this graph here showing age at maturity versus longevity that was in our reproductive resilience paper, so quite a while ago. These numbers may have changed a little bit by now. But red snapper are maturing at age 2 and living to over 50. And there's literally no other fish that I know of in the world that does that. So that would suggest to me that there's been some level of compensation. And in fact, that's what we saw with the data in the Gulf stock assessment as well. On the left, you can see age at maturity, and you can see for both the east on the left and west on the right, that going from the overfished to stabilizing time period, it's about doubled. So about from age 1, 1.5 to about 2, 2.5. And then looking at batch fecundity, and we used models based on a Clay's approach to integrating batch fecundity at length with age. You can see that it looks like batch fecundity age for those younger age groups is decreasing. And that's one of the reasons we didn't want to use that period, the most recent period, because if you use that and extrapolate it out to those ages that we didn't have, you would think that fish were doing much more poorly than they really are. Last thing in terms of red snapper, I just wanted to bring up this really nice paper that Mandy led with a great group of people. I don't know, hopefully some of you are on this call. But this idea of source and sinks, right? So we've known that fish have sources and sinks for a long time. That's been pretty well established. But we haven't really integrated that into our concept of reproductive potential. This paper showed that the biomass of red snapper off North Carolina, and that's the little star up there in the right-hand upper corner of that first map, appears to be only a sink location. That's has rather large implications for reproductive potential, right? So not all spawning sites are created equal. Spatial processes play a more important role in marine fish reproductive success than terrestrial vertebrates. I'm just going to keep hammering that point. But obviously neither SSB nor TEP would capture that. So neither spawning stock biomass nor total egg production would capture that, because we tend to think of, on the source side, so the reproductive side, we tend to think of that as constant over space and time. And then I just wanted to show on the right, again, from the same study, the center of biomass for red snapper is off the east coast of Florida. We did a study there and we're able to show that their spawning sites are really highly diverse. They just seem to spawn all over the place. And then Mandy made this nice graphic for me to the right showing what would be expected dispersal. And we're working with Mandy on a couple of things now, and I hope with her, maybe Roger and Kyle, we could discuss future collaborations on dispersal and using dispersal to do hypothesis testing, right, for nursery habitat and near class strength. So main takeaways for red snapper: reproductive compensation is occurring as the stock recovers. Reproductive systems adapt to fishing mortality either through density dependence or natural selection, i.e. fisheries induced evolution, and that's the two feedback loops we have in reproductive resilience. Given the limited age range for which we have fecundity, total egg production does not appear to accurately reflect the increased resilience and productivity of the stock as it recovers. And red snapper have sink populations resulting in spatial differences in reproductive potential not currently captured in our stock assessment. So I'm going to shift gears now and talk about sequential hermaphrodism. And this is actually the talk that Chip asked me to give because he saw the talk I gave for the Gulf SSC back in September. And I pretty much just pulled the slides from that talk, but I wanted to give a little bit of context before we get there. So protogeny is common. Protogeny is more common than protandry, which is going from males to females in fishes. And protogynous species typically live longer than gonochristic species and invest the least in male gonad mass. And that's from this really nice paper by Pla et al. And that's huge if you think about it, right? So if your oldest and largest fish are males, and those are the only males you have, and they invest the least in male gonad mass, presumably that means that you have a few males that are actually reproducing with a large number of females and producing a lot of the offspring. So it's like a fitness multiplier. But protogynous species do vary in mating strategy. We have some that form aggregations, some that are haremic, and some that are unknown. And these mating strategies, as well as other factors that we don't yet understand, will affect the overlap you see in sex-specific size and age vulnerability, sex-specific size and age, and then vulnerability to sperm limitation due to age truncation. In addition, thinking about spatial ecology, which I do a lot, I'm sure you've realized by now, with the protogynous life cycle, what it means is that all your nursery habitat and your sub-adult habitat will have nothing but females. And potentially, depending on the mating strategy, you may only have males and females together at the same time, same place on the spawning grounds during the spawning season. Age truncation is really common in fisheries, right? And it's expected to disproportionately affect males in protogynous species. But monitoring sex ratios in these species is pretty difficult, especially if they have undelimited intermixed gonads. And just in case you want to try that phrase out at a cocktail party or something and you want to learn more about it, that is also in that paper on the unified framework. But what it comes down to is fish that have that type of gonad and are protogynous, you can't tell what sex they are just from looking at the gonad macroscopically. And in all honesty, in my arrogance, because I've looked at a lot of fish gonads, I did not believe that when Gary Fitzhugh told me, but it's true, you really can't tell. So that has important implications if your fish is not dimorphic and you're trying to assess sex ratio as well. So this was the talk I mentioned that I gave back in September. And it talks about the research that we've done on gag. We started researching gag in 2015, and we've done 2 different studies on gag reproductive potential. One was the Madison Swanson study, and that sampled in Madison Swanson. That's the upper box there, just below the red star. So sampled that MPA, Madison Swanson, as well as an unprotected area that held gag and had similar habitat, that's the red star. And then the seasonally closed edges, which is south of there, and they're closed during the spawning season. And we did that study during 2016 to 2018 and had 615 samples. These studies are just hugely time demanding and stressful. So after leading a couple of these, I do understand why we haven't had this data in the past. And I'm just really impressed at the people that work with me and decide to still work with me after doing these studies, especially Hayden, who started out with the Panama one and has continued on even to our current research that we're doing now. The steamboat lumps study sampled, again, the steamboat lumps MPA and the sticky grounds to the south of it. They're called sticky grounds because they have high rugosity and not especially good for long lining or putting down fishing gear. And then some exploratory sampling off Tarpon Springs, that's near shore. And our current study is focusing more on that near shore area. And then we have great relationships with the FWC fishery dependent monitoring crew and fishery independent monitoring crew, as many people do. And we were able to get samples from them and assess them reproductively and then pooled our data with those as well. Had a master's student from FWI in the FDM section who did a really nice thesis looking at the spatiotemporal trends in gag grouper ecology as well as recreational fishing effort, which I'll talk a little bit more about. And then Hayden, who I mentioned, Hayden Menendez, is now getting her master's, and she's going to be looking at maturity in gag, doing some really interesting work, looking at new indicators and the proportion of those nearshore fish that are mature. And Hannah Gottsman, who's a PhD student, and she's taking a multidisciplinary approach to understanding population structure and using genetics and microchemistry. And dispersal. So it'll be really interesting to see. I'm really excited to see what comes out of both of these student projects. So what was known about gag ecology when we started our research? Or at least what was accepted as the gag ecology, right? So we knew they were protogynous, that they had a long pelagic larval duration and complex spatial ecology. So they spawn offshore and off the Western Gulf, that's 50 miles offshore or so, and estuarine nursery habitat. Subadult gag select nearshore habitat that has relief and exhibit strong density-dependent habitat selection. And they do exhibit ontogenic habitat shifts first from the nursery grounds, which are typically associated with seagrass, to these structure areas, and then they move further offshore as they age. Gag form pre-spawning aggregations in late fall, early winter in relatively shallow water, whereas spawning aggregations occur at 50 meters or deeper. And males remain in these deeper waters year-round, while females use shallower water and undergo spawning migrations. Sex change was believed to occur only on the spawning grounds, mediated by social interactions, so either male abundance or size of fish in the spawning aggregations, and to occur during the spawning season or just after it. And it was hypothesized, this really nice paper by Heppel et al., a modeling effort, which predicted that the MPAs would increase male sex ratio and help them recover to 15%. So what have we figured out? And you don't have to have a PhD or 30 years of research experience to probably come up with this conclusion, but it's really helpful when you're starting working on these very complex systems to collaborate with experts. And of course, fishers are experts in what's going on with fish and on the water, and they're always going to spend more time on the water than probably any scientist, certainly more than I do. I seem to spend most of my time behind a computer nowadays. As well as working with stock assessment scientists. We've been able to work with some great stock assessment scientists over the years with GAG. We held a GAG-arama meeting and brought together stock assessment scientists as well as leading scientists on GAG grouper, and that helped us get up to speed. That was early on. I think that was 2016. And then since then, we've hosted several listening and knowledge exchange workshops where we brought together fishermen with managers and research scientists and had some really helpful discussions. And I hope there'll be enough time. I'm trying to time myself and not take too long talking to have some interesting discussion after this, because I'd really like to learn more about Atlantic GAG. I had hoped I'd have time to read the stock assessment before this, but I didn't. And just hear if there's similarities or differences with what I am showing you with our system. So our conceptual model in this talk is going to draw on the one published paper. I'm way behind on publishing on the system on GAG, and the title of that is Testing Assumptions About Sex Change and Spatial Management in the Protogynous GAG Grouper, and that came out in 2020. So it doesn't include the second study, but what I'm going to present does, but in that same framework of testing assumptions. And then our conceptual model at this point is that GAG life cycle space use, their sexual system, and mating strategy all impact sex change, male recruitment, and the spatial temporal level of fishing mortality they can sustain. So as I mentioned, juveniles are in seagrass, then they move to that a little bit deeper areas, full salinity and structure. Those are all females. Then midwater, still females. Once you get past about that 50-meter mark, we found it to be 46 meters. First study we found it to be 49 meters, and then our second study we found it to be 46 meters. But still, males are only found in those deeper waters, and spawning habitat is in deeper water, with most of it occurring 65 meters or deeper. So our first hypothesis was, will GAG exhibit sex-specific habitat use? Light pink is juveniles, pink is females that are subadults or potentially mature adults, and blue is males. And just looking at that map, you can see pretty well that nursery is over the seagrass areas, and in estuaries, all the fish we've sampled there are females, and all the fish that other people have sampled there have been females. We did sample some immature females in those nearshore areas with ages 2 through 5, and they occurred in water depths less than 20 meters. Actively spawning females were documented in water depths ranging from 65 to 138 meters depth, and 138 meters depth is about as deep as we went. And most males did occur at depths greater than 50 meters. So interestingly, and again, I think this has to do with how difficult it is to sample GAG during the spawning season. So they spawn in February and March. So you can imagine what it's like making these trips out to that deep water in winter weather. So an identified need was to test whether GAG spawn south of 28 degrees. All of the prior research had been in the Madison, Swanson, and Edges area. So we have now documented spawning in Madison, Swanson, and the Edges. It had already been documented in Madison, Swanson. It hadn't been documented in the Edges, I guess. And then the second study, we also documented in Steamboat Lumps as well as the sticky grounds. That Tarpon Springs shallow water area that I talked about, we've never sampled anything spawning in shallower water. And the spawning season was the same in both, which is interesting, February 1st through April 15th. So this hypothesis about whether females exhibit spawning migrations but males do not, it looks like that's the case. So we always map out our reproductive data, and I hope this will become more and more common practice. So on the right, what you can see is a map that's grayed out a little bit, but the different colors are the different phases, how developed you are. The most important thing being orange is spawning, right? So you can see that all the spawning is in this deeper water. And then when you look at those different phases and what depths they're sampled at, and that's the graph on the bottom on the left, what you see is that you don't get spawning in the shallower water, which I've already made that comment. You don't get regressing in the shallower water either, and that's when they're beginning to end their spawning season, so that makes sense. But when you get to regenerating, these are mature females are out of the spawning season, you do get some of them in the shallower water, and the same thing with early developing. This suggests that there is some level of female spawning migrations, but looking at age and depth at capture on the right there, you can see this looks like it's mainly these younger fish, and it may be these fish that are just beginning to mature, you know, early spawners that are trying to get the hang of it, and they may be small enough that they need to come back to shallower water to forage effectively and also decrease their predation. Jury's out on that, but that's what it looks like right now. As I mentioned, measuring sex ratios is complicated, just in terms of what is a male for starters. We did find that for GAG, that male pigmentation, that really dark belly on the second from the right, was a pretty good indicator after you had training in it and knew what you were actually looking for. And if the fish had been dead, it has to be dead. So you can see that same coloring in both males and females in situ, and that's the fish underwater there on the left. We also saw less of that pigmentation further south. So it probably has something to do with the actual habitat that those fish are sitting over, and potentially doesn't occur if you're mainly over sand, like in the steamboat lumps. Again, complete hypothesis. But the accuracy was 90%, which is really quite good if you have the training, but hard if you have fisheries dependent sampling and a high turnover, because it's difficult for those people to get the training, and then often they don't stay long term to be accurately assigning that. So in GAG, they exhibit sexual segregation, which I showed in that map with the pink and blue. And it means that mature individuals of both sexes are only occurring on the spawning grounds during the spawning season. So that's when you need to estimate your sex ratios. But it also means that a number of things will impact your sex ratios, even if you sample them during that time and at the location. One of those is a strong year class. So anything coming in as a strong year class is going to be female and will make it look like you have fewer males. Or if there's skip spawning, and in this case, I don't mean not developing. We did see a lot of that, but I mean fish that actually don't move to the spawning grounds with variable participation in spawning migrations, then you would also have a different sex ratio. So it was hypothesized that the MPAs would protect male age structure, and it looks like they did. Madison Swanson age at transition, so that's the A50 there, was 13 years. Steamboat lumps the same. And the pooled data was 11.6 years. And if you remove the MPA data, it's 10.4 years. But interestingly, this is really similar to what's been seen historically, both when the stock was considered to be doing much better, 1977, and then more recently when it was considered overfished. And the A50 at that time, or at least what was used in the stock assessments, was 10.9 years. As I said, it was hypothesized that not only would it protect male age structure, but it would allow male sex ratios to recover to 15%. This is not what we saw. Both MPAs had male sex ratios of approximately 5%. And then unprotected areas showed very different male sex ratios. In the edges, we didn't catch any males during the spawning season. We did catch some males outside the spawning season, and males do occur there. But apparently in low proportions. Although longliners told me that they could catch a lot more than what we saw. So take that with a grain of salt in terms of we were sampling with chartered fishermen and doing hook and line. And then sticky grounds actually had higher male sex ratio than the MPAs. But again, that's way offshore and in an area where it's not easy to set fishing gear. So putting these sex ratios in context, overall, it looks like the male GAG sex ratio in the Gulf is quite poor. CEDAR 72 estimated at less than 2% based on an A50 of 11.6 years and the age comps. They estimated virgin sex ratio to be 32%, which is important. It does not look like it is the reproductive strategy of GAG to have ever been 50% male. And previous studies found very similar low sex ratios. So Koenig and Coleman 2011, 5% in the Madison-Swanson MPA and only 1.1% outside. Burns and Robbins, again, small sample sizes, but 1.8%. So it's looking like about 2% currently, male sex ratio, compared to historically, and again, some questions about how it was all measured, but historically it was estimated to be 17%. So the fact that the MPAs protected male age structure, but did not allow male sex ratios to recover, of course, makes one scratch their head about what's going on with male recruitment, and is poor male recruitment the problem? So what sorts of things affect sex change? This is not especially well known in anything other than blue-headed rats, but the main theoretical model is the size advantage model, and it predicts that sex change evolves when larger age or size of the terminal sex results in greater fitness, and that makes sense. So again, the theory that if your reproductive success significantly increases with size and age, you want to change sex. It's tightly linked to the species mating system and social structure. And the known triggers and mediators in these systems are population density, local sex ratio, relative size to others within the social group, and potentially a threshold size or age. So starting with the mating strategy, it has been reported and published that GAG forms spawning aggregations. It was always reported that these were rather small, 50 to 100 fish. Based on recent definitions, quantitative definitions by Daumier et al. Of a fourfold increase in density to define spawning aggregations. So you have to have fourfold increase in density during the spawning season versus outside of the spawning season. Neither of our studies supported that with our various measures, either hook and line or video. And the most we had seen, the highest number of fish we had seen, GAG, in our videos was 12 in steamboat lumps and 18 in Madison-Swanson. So when we think of spawning aggregations, we often think of Nassau grouper, this beautiful picture here on top. But what we were seeing with GAG was a very different story, never large numbers, and also they're pretty camera shy. But then, there's always the issue of studying a system more than 3, and in this case, 6 years. You're bound to find something that completely messes with the original results that you, of course, already published. So last year, this is a site in steamboat lumps that we've consistently assessed, and in our current research, we're actually using ROV surveys to look at months in the pre-spawning time period versus spawning time period. And we saw 42 GAG at this site. And if you look at those little bar graphs to the right, red is spawning, blue is pre-spawning, that would actually meet that quantitative criterion. That's just to show that this is actually video. And the dark belly you can see on that one there, that could be a male or a female. So again, the hypothesis was that sex change would only occur or primarily occur on the spawning grounds. That's not what we're finding. We're actually finding sex change in shallow water is common or more common than on the spawning grounds. And what that means is that males are not needed. It's not a low male sex ratio, well, a very low male sex ratio, I guess. You don't have to have males. It seems to be female-female agonistic behavior can be a sex change cue. Those that were changing sex in shallow water were smaller and younger than those on the spawning grounds, which has important implications for adaptability and suggests that the size and age of GAG sex change is dependent on the relative size of others in the social group. So for GAG, we don't really have big old fat fecund fertile females, but we do have big old males or bombs. So GAG males are significantly larger than females, and in the graph on the right, that's the blue dots versus the red dots, so blue is guys. And you can see that they are not the fattest. Our biggest fish actually was a female at about 50 pounds. The largest males we sampled, they were both sampled in Madison-Swanson, and they were 40 pounds. But that's not nearly as big as males used to be. And you can see that picture of a 72-pound male captured in 1985. And so if you think about size and sex change theory and the relative role of bombs, big old males, it would suggest that we have lost a fair amount of reproductive potential in this stock. So another hypothesis, and this was not in that paper, although I guess this was discussed, is this idea that we often think that protogynous species will show similar patterns, especially if they have similar mating strategies. Although in this case of gag and scamp, the mating strategy really isn't known for either. And you can see they don't. So the gag are on top, and you can see very few males. You can see much higher females and at younger ages. Whereas with scamp, you have a lot more males, a lot more overlap, and much higher numbers of transitionals. So gag, we rarely sample transitionals. Less than 1% of all of our samples have been transitionals. But interestingly, gag and scamp live to approximately the same age, about 33, 34, and they both mature at a similar age, 3 for scamp and 4 in gag. But again, you can see the age truncation appears to be worse for gag, and you'd expect that. There's a lot more fishing pressure on that stock than on scamp in the Gulf. And again, we don't know what their mating strategy is. We simply know that they're not haremic, but we do know that both of them appear to be pair spawners, and they have very little milt reserves. So again, before I started working on reef fish, I worked a lot on nearshore fish that have sperm competition, spotted sea trout, snook, and red drum. The males all have very large testes, and they produce milt. You could always tell what a male was, but that's not the case with scamp and gag. And in fact, we've sampled gag during the spawning season. You could not actually express milt, or even if you cut the testes in half, you could not see milt. You actually had to do histology to see whether it was a male or female. So very low milt reserves in these species. So in terms of their spatial ecology, how similar it is, we have sampled spawning females of both gag and scamp in those 2 studies on gag in those areas. And we commonly see courtship color changes and behavior in scamp. And I had some fun videos, but I didn't have time to put them in here, but you guys probably already all seen fun videos. Gag have estuary nursery grounds. The nursery grounds for scamp are actually unknown, but presumably in deeper water. And then I mentioned that student of mine, Rachel, who did this nice thesis and was actually able to show that 94% of sampled recreational fishing effort was in less than 50 meters. At the time, we thought that's the main cutoff between males and females. So mainly on females, recreational effort is mainly on females. And that there is increased catchability in the fall in those shallow water areas. Our ongoing research, I'm really not going to talk about, although I'm really excited about it. We are using a number of ways to sample nearshore gag and also acoustic telemetry in the hopes that we'll be able to track some of these movements and also be able to evaluate what are the triggers that cause mature females or females that are maturing to leave these nearshore areas and move off to the spawning grounds. So main takeaways for gag and scamp. Gag male sex ratios in the Gulf are low, and it takes about a decade to make a male. They did not recover to the expected 15% in either MPA. There is intense fishing on the females in relatively shallow waters, and this may cause a bottleneck to spawning population recruitment. Really male recruitment, you just may not have enough, well, both, I guess. You may not have enough females that are maturing and getting offshore to spawn, and you may be killing some of those young transitionals before they can get a chance to get off and get offshore and help replenish the male population. But there's not a lot of targeted research on scamp. We currently have a proposal in, hopefully that'll get funded because we're really interested in better understanding that system and comparing it to gag in the hope that it'll help us improve our understanding of protogynous species and how best to manage them. Okay, so wrapping up, the whole idea of reproductive resilience is to take this systems approach to understanding multi-generational productivity, and this means often doing comparing across species, and I would say that gag are more like orchids and red snapper more like weeds. And isn't it interesting? We have those ideas in plants. I don't think we have. Well, I guess we do in animals because we think about rabbits, but they only produce 84 offspring a year. So neither gag nor red snapper appear to have recruitment closely tied to SSB or TEP. They have different sexual systems, and each truncation is expected to have a much greater impact on gag than on red snapper. Female maturation in red snapper adapted to fishing pressure. Maturation in gag, male gag, does not seem to have done the same, right? So that was that A50 of about 10.9 throughout time. Gag have estuary, nursery, and offshore spawning habitat, whereas red snapper spend their life cycle offshore, so not as large of a spatial footprint. They do not appear to aggregate at any phase, whereas GAG appear to at least clump up in those nearshore areas in the fall when cold fronts go through. And red snapper do not appear to exhibit spawning site selection, whereas GAG have specific spawning habitat. So red snapper have this really large diversity of spawning habitat and appear to be able to produce offspring that survive and appear to be incredibly adaptive. They also can produce these really small batches, which again, I've never seen in any other species. So I would consider them a weed. They seem very resilient. But then also looking at species across time and space. So thinking about reproductive compensation versus fisheries-induced evolution, red snapper are kind of our cod. There was a nice paper that just came out by Pinsky showing that in cod, in terms of genetic diversity, it was not compromised by the overfishing. So I think these large marine populations often can recover, and reproductive compensation may turn out to be more common than we had thought. Obviously, in terms of climate change and changing oceans, a big issue is going to be whether you have a disconnect between spawning site selection, the cues that drive fish to spawn at a given site, and if those are constant, while oceanographic conditions change, you're going to have different recruitment success. Then I'm really fascinated by these species that on the East Coast are doing fine or not doing fine, and in the Gulf just the opposite. So hopefully we'll get a chance to talk a little bit more about those, or I'll get a chance to learn a little more about those, because I think that's fascinating, and whether that's because of the oceanographic habitat system or how we sample them or something else is an interesting thing to be working on. So main takeaways for reproductive resilience and in terms of management, there's not going to be any time soon that we're going to have all this data for reproductive resilience for multiple species. There was this nice paper that Andre led in 2022 for small pelagics, and he did used experts on pelagics to ask these questions, a questionnaire on demographic growth rate and larval dispersal, recruitment variability, reproductive effort, all those things that are in the reproductive resilience paradigm, and was able to look at how that differed across the species. But it's not like we're going to get hard data for those things anytime soon. But as a concept, it's already changing our thinking about recruitment processes in marine fish, and I think that's really important. And like dispersal models and integrated life cycle models, reproductive trait space is not part of stock assessments, but I think can help us better understand resilience. With that, I just want to thank all the fishermen, all the experts, and especially the people in my lab who have worked with me, a lot of them over many, many years under pretty we tend to take on really hard projects. So with that, I'd be happy to take any questions people have.
All right, thanks, Sue. Great presentation. We're going to open this up for the question and answers. I'm going to go ahead and take that control. Okay, so just as a recap and refresher as well, you can ask your questions two different ways. You can type a question into the text box below on your console. If you'd like to ask it via voice, you can raise your hand. I'll keep a list of the hands raised, and then in order I will unmute you and you can give your question. And to kick us off, Sue, I've got a written question coming in from Ashley Weschler. It says, great talk, thank you. If you are calculating spawning fraction, but using all fish in oocyte maturation, could you change your correction factor, i.e. not divide by 1 for the frequency?
So great question. That depends on how long oocyte maturation takes in your species. And the only way to really know that is by doing in captivity studies, which most of us don't do. Or if you have a species that has really strong deal periodicity, you can figure that out by collecting samples over times, right? Maybe over 2-hour increments, and then looking at time between when you get that early germinal vesicle migration versus when you get full hydration. Did that answer your question?
I don't see her raising her hand. Yes, thank you. She responded. Okay, any other questions? Please raise your hands. Got Kyle Schertzer. Go ahead, Kyle.
Hi, Sue. That was a really nice summary of a huge body of work that you've been involved in. Early in the talk, you made the case that spawning biomass may not be a good proxy for population fecundity. I'm curious if you have any thoughts on how a population model or a stock assessment model might use fecundity for protogynous stocks in a way that could account for the importance of males.
Okay, I think we've had some of this discussion sometimes with SCAMP, but so yes, there are ways to measure sperm production similar to oocyte production. They typically use that in things like racehorses. So that is something that could be done. I think the bigger issue is that fecundity just by itself, whether it's male or female fecundity, is probably not the driving factor, right? You have so much offspring mortality. So it's where and when those fertilized eggs get released, as well as how many are released. So I think in terms of thinking about how to weight reproductive value at age for males or for males in general, it certainly depends on what level of overlap you have in size and age, like we saw with SCAMP versus GAG. I'm much more worried about age truncation in GAG than I am in SCAMP or red grouper. And that's going to depend on what drives sex change in that particular case, right? So GAG, for whatever reason, are not adapting the way some of the other species seem to be adapting to fishing mortality. So in terms of how to weight males, I think Nikolai did some of this for his PhD. I do think looking at male SPR is probably a good idea. And that's why I'm pretty excited about working with the stock assessment scientists and looking at virgin, what would be expected virgin sex ratio. And that's probably the best we have right now. I do think we need to also do a better job probably. Well, it's very hard to do anything in captivity. They have gotten GAG to spawn in captivity. But of course, as soon as you do something in captivity, it's hard to, especially for something that's spawning at 50 meters, to extrapolate what you see in a tank that's maybe, 3 meters deep to what happens in the field. So right now, I think the best we can do is probably male SPR. What do you think?
I don't know. I've been thinking about it for more than 20 years and I don't have a great answer yet. But somehow I feel like we need to include egg production, but try to model sperm limitation in some way. But quantifying that is really hard.
Yeah, so I guess that you would like to see a way that we could actually literally quantify sperm limitation, right? And we've done some back of the envelope stuff with males, looking at so if you have 2% male GAG, you know what the fecundity of your females are, and you know what the sex ratio is, you know how many females the male would have to fertilize. You could do something like that and get a threshold. Again, you're gonna have to use spawning frequency, so there's a lot of uncertainty there, but you could look at how many eggs each male would potentially have to fertilize, and then do some in situ studies in terms of sperm production to see if they can actually do that or how far they fall below that. I think that would be cool and helpful.
I agree.
And I also think, a lot, I just sat on this review panel that was like a little primer on all the latest genetics techniques that are out there. And and they're doing some really cool stuff. A lot of it was European. And I do think there will be more and more things coming along in relatively rapid time that could begin to help us understand that. One of the things they were talking about was identical birth, or the IBDs, identical birth, can't remember what D stands for, but these really long strings of genetics that can help us, that totally change our ability to understand dispersal and reproductive success. So I do think there'll be things like that coming along soon. I'm pretty excited about the CKMR stuff as well, because then these really large genetic samples are being collected and analyzed, and hopefully that same data can be analyzed for reproductive success in some of these processes.
Yeah, well, thank you, Sue.
Okay, next up we have Cameron Sebastian. Go ahead, Cameron, you're unmuted. Cameron, you might be muted on your end.
Yeah, so the question was, I believe in one of your things you were saying that you saw you used hook and line and video to collect some of the data. Is the video all used on an ROV, or is it used on ROV and stationary GoPros on traps, etc.?
So the first two studies we did on GAG, we had a stationary, we had GoPros on a stationary base that went to the bottom, had 3 GoPros, so you had close to a 360 view of what was going on. But at these depths, you get a lot of current at the bottom, and that impacted what we were getting to see and whether the system actually moved and whether you could get on the spot you really wanted to get on. So our most recent study, we now use the ROVs, again, with 3 GoPros, but so you can actually motor and set your cameras where you need them to go.
So are the GoPros mounted on the ROV, or does the ROV have a camera mounted system on it? Or are you just setting the GoPros with an ROV?
The GoPros are mounted on the ROV.
Gotcha. All right, thank you very much.
Okay, any other questions for Sue?
Hey, Sue, this is Chip Collier with council staff. Sorry, I couldn't raise my hand, but I'm just wondering, with some of the terrestrial ecosystems and some of the terrestrial species, they typically follow a typical spawning route, and sometimes it's really up to the older individuals to show the younger individuals the route. I'm curious if you've seen that with any of these fish species, where the older fish tend to lead the younger to the important spawning locations.
Yeah, great question. So I know there are a number of papers on that, or at least hypothesizing that. We don't have data that would let us actually test that hypothesis because of the time span and life stage span. You'd have to electronically tag fish. But with that red drum, that extensive red drum study we've done, I would suggest that anything that schools like that with a separate ontogenic habitat for juveniles probably does. So we see those really large spawning aggregations. I guess I didn't show any pictures in this one, but they're really cool. These beautiful, huge spawning aggregations that come and spawn off of Tampa Bay. And they move into the mouth of the bay, and that's where the subadults move down to during the spawning season. And that seems to result in the recruitment process of the fish that are maturing into the adult population. And then because they school, they have the older fish, and they had this very tight, very strong schooling behavior. So the way we sampled them was with purse seine. As I mentioned, it was non-lethal, but you would release the fish. So we'd hold them in the net, the ones that were sampling up, and then release them, and you'd see the fish they'd been released, and they'd swim around and just stay on the outside of the net waiting for their schoolmates. So I would think in that system, certainly the older fish are going to be the ones that are presumably, leading, and you see that with birds as well. So I would expect that to be the thing.
Thank you.
All right, next up, I've got Jeff Buckle. Oop, you just left. Oh, sorry, Jeff. Hold on a second. Let me get you unmuted. All right, go ahead, Jeff.
Thanks, Jed. Sue, great talk. Really impressive amount of work. I'm curious on the, you talked about the gag not adapting, and it sounds like that, but red snapper have to fishing, and you mentioned density dependence as well as fishery induced evolution. I was curious if on the red snapper, which of those do you think's at play? I know that sounded like an area for future work to try to disentangle those two, but do you have any indication which is at play for red snapper?
Well, based on what we're seeing in terms of the compensation and the age at maturity increasing, I think that that's going to be a reproductive compensation issue rather than a fisheries induced issue. I wouldn't have, if you'd asked me that 3 years ago, I wouldn't have had that same answer, but it is really, I was just thrilled and amazed to see the changes over time with those 3 different stock periods for red snapper, both with maturity and with batch fecundity. So it's looking like that will be the case. And like I said, there's that nice paper by Pinsky that just, I think it came out a year ago on Northeast cod, which suggests similar. And I think like the effective breeding versus census population size ratios that were used for that sweepstakes reproductive success hypothesis, I think, you know, we're just, again, on the cusp of really learning how these large marine populations work, and that they work very differently than what we've seen in terrestrial systems with low fecundity. So I think it may turn out that a lot of fish species actually have more compensatory, that even with the high fishing mortality and decrease in age structure, if you release that fishing mortality, they may have more potential to recover than we would have thought. I always made the point that if you kill off all the old fish and you select for fish to mature earlier, then you're going to have smaller fish, right? Because a lot of the energy is going into reproduction. And potentially you've just decreased the productivity of that species. I think the jury's still out how common fisheries induced mortality will be, but the limited data we have on red snapper, I'm thinking it's looking like they're going to be reproductive. It's going to be reproductive compensation, if I could say the phrase. But that's what I think is going on.
Thanks, Sue. And yeah, great talk. Enjoyed it.
Thank you.
Hey, do we have any other questions for Sue? Okay, I've got Myra Brower. Go ahead, Myra, you're unmuted.
Thanks, Jed, and thank you, Sue. So my question is, this kind of brought me back to my days doing my research in grad school, where I spent lots of hours under a dissecting microscope with a little counter counting hydrated oocytes, and lots of hours under the microscope looking at those POFs. So I'm curious what is going on these days in terms of technology to make that job a little easier, faster, maybe like AI technology. I don't know. How do you all do that?
Great question. Well, so the Panama City Lab has been doing a lot in terms of, we've been collaborating with them, a lot of things that they're integrating into their age and growth. We're also seeing about integrating into reproduction. So I think a lot of it is moving towards more quantitative histology, right? Image Pro and writing macros that can quantify what's going on there. So you're not doing that manually, whether it's hydrated oocytes or histological indicators. And then one of the things that's going to be a game changer are these histo scanners that they're actually developed for pathology labs, where you can scan your slides at multiple levels of focus, and you can share them. So one of the big things with reproduction is that the things that have occurred in age and growth, to try and standardize methodology are just beginning to happen in histology and reproduction, but that's because the technology is just becoming available. But so those are some of the things that we're doing. We're probably not the leading, I don't know, we're trying to catch up on the leading edge in terms of some of those reproductive things. We have been spending a lot of time working on movement ecology and all those arrays, and what it takes to maintain all those arrays is a huge sink in terms of the lab. So we're just now, and Hayden is doing a lot of that with her master's, trying to get up to speed on some of the quantitative histological approaches. But it'll be really nice, even just the Image Pro and being able to count your eggs without doing that manually. Because like you, that's what I did as a graduate student as well. And let's save some people's eyes.
Okay, I've got Chip Collier with another question.
Yeah, during your presentation, you had mapped out a pretty small area where gag grouper spawn pretty much off the western shelf of Florida, the northwestern shelf. And I'm just curious if on the South Atlantic side, if spawning is believed to occur along the coast, or is it just a few areas? I know Oculina Bank is one area where spawning groups have been observed, but I'm curious if they're seen down in the Keys or even further up off the Carolinas. So we have people, not only yourself that is an expert on reproduction, but we have several fishermen that might have input, as well as other scientists, on whether or not they're seeing spawning groups of gag off the East Coast.
Yeah, I'd love to hear that. On the West Coast, they think that those are, you can see these drowned riverbeds. They're very specific geological features that seem to attract where they spawn. I know there are gag in the Keys. Are there some people on that can discuss that a little bit? I'd love to hear more about that. Are you out there?
Not seeing anyone willing to come forward with a response right now, but if someone does have some information, we can pass that on to Sue if you'd like to send it on to the council staff.
Is Kyle still on? Because I know in the past, talking with Dave Wayansky, it did not seem like they, or Maya, maybe whether you all map your, when you get spawning fish, do you map out where they come from? Do we have data like that on the East Coast? Kyle probably had to go to another meeting. Oh, well.
I'm still on. There's nothing that I'm aware of, Sue.
Oh, okay.
Kevin Colmos has his hand up.
Hey, can you hear me?
Yeah.
Yes.
Great. Yeah, I mean, we do have the information of where our spawning fish come from, but I don't think we've actually mapped it out at this point, but it's definitely something that we could do with a number of different species. So we have the data to do it. I think it would be pretty interesting to see, especially since you're seeing such a like the sexual segregation by depth. I think that would be pretty interesting to see if we are seeing the same thing on our side of Florida and on this coast.
Yeah, that would be fascinating. And do you know if do you have the same fall nearshore fishery that's really intense on relatively young gag on the East Coast? Or is there a fisherman who can answer that question for me?
Yeah, I wouldn't say I know that off the top of my head.
I've got Cameron. You want to try to answer that question for Sue?
So, we run charter boats out of the borderline of North and South Carolina, and we actually have seen a large increase in juvenile gags and keeper gags in shallower waters than we've ever seen in the past 35 years that I've been doing this. About a month ago, I was diving at only 7 miles out which you never see gags of any quantity, you know, a few here and there were at least 5 to 6 keepers with 15 to 20 juveniles in just one area, which is absolutely unheard of, or I've never seen it in the 35 years I've been doing it.
Wow, that's really interesting. So we also had a really good year class. Right now, there are juvenile gag all over the place. The most we've ever seen. And as I showed on that video, so the year before, we saw the most spawning gag we've ever seen. So it does make one wonder what triggers gag to spawn. Before we started doing research on gag, there was the dogma that you get a strong year class every 6 years or so, and that was, people didn't quite understand what drove that, but you would get that. And certainly this year we just have tons of juveniles. But so do you get juvenile gag in your estuary?
We see some small ones. I mean, the guys who they'll cast nets for shrimp and stuff like that, and they'll find some juveniles in there. I was referring to the nearshore water between 1 and 15, 20 miles out. We've definitely seen an increase in the numbers of gags in that area in the last several years. It seems like they're, and I don't know if anybody can answer that question, is, you know, do they migrate in like that? Because like I said, it was almost mind-boggling to me to see that many gags at basically 6 miles off the coastline, and I've just never seen it before in our area ever.
Oh, okay. That's really fascinating. So I'm gonna, hopefully there's somebody out there that can answer that question for you, Cameron. On the west coast of Florida, certainly in the Big Bend area, we get really large numbers of sub-adult gags, so like 2 to 5 year olds in those nearshore waters, and that's and the best fishing, and it's everybody knows it, it's online, you know, is in the fall when cold spells go through. So yeah, that'd be really interesting to understand. Can somebody answer Cameron's question? I'd love to hear the answer.
We've got Wally, Bubbly, and then Kelvin Colmos perhaps to that question. Go ahead, Wally.
Yeah, I don't know if I can answer exactly what Cameron was asking about, but I mean, just backing up what he was saying as well is, so we were getting anecdotal evidence of a big gag year class in the estuaries around us, and so we had to really twist some arms and get some people out and go to the point right by our lab and just do some fishing once a month. And at one point, I think they caught, in the span of a couple hours, they caught 15 or 19 small hand-sized gag that were young of the year. So, I mean, we're seeing a big year class like you had mentioned and Cameron had mentioned as well, moving through, and then they tend to move obviously offshore as they get older, and hopefully we'll see them in the traps and show up on the videos a lot more in the years to come.
But isn't that fascinating that the strong year class for gag would happen on both coasts? There's pretty different oceanographic conditions, I would think. That's really cool.
Yeah, and this is Chip Collier with council staff. Julia Bird had also noted in release through some of our citizen science data that they were seeing a pulse of small gag in the reports. Julia, if I'm wrong on that, you can speak up.
Julia, go ahead to that.
Yeah, I was just going to say that's right, Chip. It was in, I think it was 2022 when we saw more people were seeing those kind of hand-sized gag inshore. And then like what Cameron was saying too, this past year, it seems like those really kind of smaller young of the year fish have moved off to kind of nearshore waters, shallower and closer to shore than they have been seen by kind of fishermen that we've been talking to and have been fishing a long time. So it seems like, you know, that good year class is slowly moving kind of offshore just from kind of information that has been logged in kind of our release project and from talking to some of the fishermen who are participating in the project.
Thank you.
Great, thanks,
Really
Julia.
cool.
Got Kevin Colmos and then Jack Cox and then Steve Turner.
Yeah, just kind of add on to what Wally was saying. We saw, I think that information was kind of from last year, and then this year when we opportunistically went out there, you know, we weren't catching much at all. So I don't know if it's a temperature thing, if it was just a strong year class or what, but there was pretty big variations in what we were catching. I also service some of the VEMCO receivers that we have offshore here and along the channel, the Charleston Harbor, and just diving on some of the sites that are probably within that 7-8 mile range. There were a lot of those juvenile gag out there as well. So, you know, hopefully we're continuing to see this trend of smaller gag being recruited into the population. So again, just kind of more observation than you know, standardized data collection, but good to see.
Yeah, it sounds like maybe the strong year class on the East Coast was 1 year prior to ours.
Yeah.
So that, those are the things with dispersal modeling that I really hope, Kyle and Roger, if you're out there, hopefully we can talk. I really hope that we can look at those. GAG would be a perfect case study for that.
Next up, I've got Jack Cox. Go ahead, Jack.
Yeah, hey, Sue, thank you for your presentation. I enjoyed it. Snapper Grouper AP member, and I've spent a lifetime off of Beaufort, North Carolina, commercial fishing for gags. And part of your presentation was showing in the MPA areas that there is a higher number of gag males. I'm correct on your presentation. But I just wanted to say, over my career of 40 years of fishing for gags, that off the shoals of North Carolina, in about 40 to 50 feet of water, back in the 90s, early 2000s, there was a tremendous amount of larger males, and they were very. It was during April and May that we would see the abundance of fish, and I was pretty sure those fish were in there spawning. I did a lot of diving in the area at the time, and of course, fishing pressure has dramatically decreased that population of fish. But yeah, I'm very interested in some of the MPA work that can be done, and to rebuild this fishery that I have seen over the years decline, and would love to see this fishery rebuild to what the red snapper fishery's done. And I think I was hoping to see that the council maybe on the East Coast pushed that spawning season out through May, because we were noticing back in the day that those fish were heavy and eggs and stuff, and early May. But a lot of this research looks like you've done has been done on the West Coast, down into Florida. What similarities are you seeing on the East Coast, or has it been very much work done in comparison to what's been done over there in Florida on the West Coast?
Yeah, that's a great question. I have not actually, I've worked on red snapper on the East Coast, but I have not worked on gag grouper on the East Coast. I think that would be a great thing to think about, is setting up some research that is similar so that we can use the same methods so we can actually make those comparisons. It is, when you see those historic pictures of those really large males that, they do have the copper bellies, you can tell they're males and they're huge, right? And we didn't see anything like that even in the MPA, right? So we're seeing about half the size of what recreational fishermen, in terms of males, we're seeing the maximum size about half the size of what they used to be able to catch in the 70s and 80s in the MPA. So it is a little shocking, and I'm sure there's multiple issues at play, but certainly part of it on the West Coast anyway is that you can now have your go fast boats with your, four engines. So things that used to be spatial refuges do not seem to be quite as much spatial refuges as they used to be. But that's really fascinating. So, and if you have any old photos of gag, or, I'm really fascinated by that. I do think that we do have this idea of shifting baselines, and people get used to what they think of as big gag, and they're not even close to what used to be big gag. So especially with a species like gag, if in fact they were changing sex at the same age, so around that 10 to 11 years old, even when you had those big gag that lived much longer, we have seriously depleted their reproductive potential. happy to send you my email if you have anything else you'd like to share. If you have any photos, I'd love to see them.
Okay, I'll see what I can pull together for you. Thank you.
Thank you.
Next up, we have Steve Turner.
I'm going to ask about density dependence. Basically, there was an assessment for Gulf Gag, and the assessment was done in probably the early mid 2000s. It showed a substantial increase from the late 80s, early 90s to 2000 or later. And it seemed to me that there was a periodicity in recruitment. And I'm wondering, on the order of 3 or 4 years between peaks. And I was wondering if anybody has seen density dependent effects on abundance or recruitment, and or has anybody observed in food habits or et cetera, predation, cannibalism. Anyway, thank you.
Well, I do know that they can be cannibalistic, that has been observed. I don't know that's from working with fishermen and then sending me pictures, as opposed to having scientific data on how commonly that happens. You're probably talking about CDAR 33, maybe. The thing is, that was based just on female spawning stock biomass. Although if what you're talking about is the pattern of the recruitment, that wouldn't be. And my understanding is that on the West Coast, that was before my time, that they used to see, before I started researching gag, that they used to see strong year classes every 6 years or so. But then that they hadn't, so we are seeing a strong year class right now, but this is the first one we have seen since I started working in 2015, and I think it's about 5 years before that as well. And so for the interim assessment, because we're in a recovery plan right now, they did present what looks like the resilience pattern, the recruitment pattern. We're still below what would be mean recruitment. But I think once they get the data from this year, that will change. Does that answer your question? In terms of density dependence, obviously density dependence happens, and seeing how many little gag are out there, it has to be happening to them. That's one of the things about fisheries and their reproductive systems that often isn't taken into consideration, is that this overabundance of offspring means that is food for somebody else, whether it's eggs and planktivores or whether that's fish feeding on juveniles, including cannibalistically, that energy is going into the system in a different way than it does in terrestrial systems.
Thank you for that response. That's interesting. I hadn't thought about other animals focusing on gag when they're abundant. That's an interesting comment. Thank you.
Julia?
Hey, Sue, this is Julia with the South Atlantic Council. One of the things that you just mentioned about having old historic fishing photos, it may be worthwhile connecting with you offline, but one of our citizen science projects called Fishery is kind of gathering old historic fishing photos from kind of the South Atlantic region, from in particular the for hire sector, to get a better idea of kind of catch and size composition before catch monitoring programs were in place. And we have pictures that definitely have grouper and some gag grouper. Sometimes the grouper can be hard to identify the species in some of these old black and white photos, but if that's something you're interested in learning more about, maybe we can connect offline.
Yeah, that'd be great. I'd love to.
I've got one more, Matt Vincent, and then we're going to wrap up. Go ahead, Matt.
Hi, Sue. Thanks for the great presentation. I had a question about the spawner frequency. So that's all based on a calculation of looking at the histology. Has there ever been any studies that do like some sort of tagging or some sort of visual observation of like individual fish to see how accurate these fish are, like actually spawning multiple times? And are there any ways to actually do that, or is it just impractical?
Yeah, great question. So, I mean, obviously you can do that in captivity, although I'm not aware of people actually doing that. And then we have done that with spotted sea trout and red drum and snook. So with spotted sea trout, we just had this ideal situation. Because they make courtship sound, and they spend their whole life cycle in the estuary, we could map out all their spawning sites. We mapped out all their spawning sites in Tampa Bay. And then we found 2 reproductive behaviors. One where fish were spawning in the same areas they were year round, and their home ranges where they're foraging presumably, and relatively low spawning fraction there from 16 to 26, I think it was. That was a slide I had, but I took it out. But there's a paper on that if you want to go find it. And then this site at a past spawning site that fish were moving to spawn. So we sampled fish there. We would only catch them in the evening when they spawn and during the spawning season. And that was the site that had 97% of the females sampled there were spawning, right? So we set up an acoustic telemetry study, a couple of them actually, and looked at fish moving to that site and used that individuals over time, and used, if you're at the spawning site, to estimate spawning fraction and frequency. Now, the caveat being, I can't tell you that fish didn't go and spawn somewhere else as well. We did have fish that showed, spawning site by LE to that site, moved to that site repeatedly multiple times. And it was nice because you then got males and female spawning frequency, which we don't usually get. Males spawn much more frequently. On average, they are moving there every 2.2 days, and females were moving there every 9.3 days. Females came from farther away distances, which is kind of interesting. So you can use, and there have been some other studies as well with that type of electronic tracking. And then there have been a few individual based studies on things like seahorses and some other species with very specific reproductive strategies that it's easier to look at the individual. They never agree with the spawning fraction is the take home message. But that hasn't been very, I'm not sure that case has been made strongly enough that you're basically, you cannot get an individual estimate of fecundity in these fish that have indeterminate fecundity. You have to get batch fecundity and then your best estimate based on spawning fraction. And the few studies that are out there suggest that spawning fraction can be off by quite a bit.
And we've got one more, Will Heyman. You're unmuted, go ahead.
Will, are you out there?
I'm on a phone and I hit the wrong button. Hopefully you can hear me now.
I got you now.
Well, thanks, Sue. Great talk and a great series of questions. One quick thing that I wanted to point out in terms of historical photos, and I think I sent these along, but if I didn't, let me send them again to you, Julia. But some amazing old video that was taken in the 80s and 90s on some gag and scamp aggregations that I ran across. Chris Koenig sent me. So let me get that over to you guys to make sure people see that, because it is astonishing, the numbers of fish and the sizes of fish. Some of this was in Oculina banks or whatever, but I think that is worthwhile reminding everybody that yes, just because we don't see big aggregations of these things anymore, and doesn't mean that they never existed and may come back again. And in that regard, I want to kind of big up the South Atlantic Council in terms of making spawning special management zones to protect multiple species, including gag and scamp, and just let you know that we will be indeed out in Georgetown Hole this spring. We did document some large gags out there last year, and we'll be trying again to see if we can see that MPA Georgetown Hole is indeed holding those reproducing males too. So, and then Yeah, so that's it. I just wanted to, no, the last point that I wanted to make was simply that, and it was a question that you had asked earlier, Sue, regarding locations of spawning fish, and I just wanted to point out to the MARMAP database where, you know, all of those fish that those guys document up and down the East Coast, that data is available, and the review paper that Nick Farmer did on the East Coast with several of us on the call pointed to that. And a lot of those sites where we're seeing gag spawning, we're seeing scamp spawning, and we're seeing multiple species spawning. So that's just a reminder that MPAs, you know, can indeed hopefully protect multiple different species to spawn. So thanks. Sorry to speak so long.
Thanks, Will. So I'm on the Nick Farmer paper as well. And I think that one of the things that hopefully Kevin, you know, we can discuss is doing more with the spatial mapping of reproductive phases. I think one of the big things that Nick encountered is that he didn't have as much data on the spatial reproduction as he'd hoped for. And then in terms of those videos, yeah, Will, those videos are really cool. So, you know, Chris Koenig is a co-author and co-PI on our studies, and he shared those videos with us as well. So happy, I think we digitized ours. I think he gave us the original tapes actually, but it is really impressive to see, but it's also really good, I think, to see that even at that time, it was smaller numbers. And I should also say that for scamp, we have seen what looked like a spawning aggregation of scamp in Madison Swanson. We didn't see that with gag, but we did see that at this one site, very large numbers of scamp. Again, our videos are on the bottom. I think most people's are. So if you have a lot of spawning activity going on up in the water column, you're probably not going to catch it. We've tried our best with gag. We've tried to be our best voyeurs. We've tried different depths. We've tried everything, and we couldn't get something. But anyway, those are really good points, Will. And yes, I think in terms of MPAs, I am not by any means saying that those MPAs did not work. They simply didn't result in the expected recovery of male sex ratio, but in part that's probably the gag spatial ecology. So red grouper in steamboat lumps is huge, and steamboat lumps has been very effective at protecting red grouper, but they have a different spatial ecology, and they don't have those nearshore estuarine nursery habitats, and they don't have that really intensive fishing on nearshore subadults.
Great.
Well,
All right. Well,
again, thanks. Yep, I'm done.
Oh, okay. Thanks, Will. All right. Well, that's going to wrap us up. Just like to say thank you, big thank you to Sue for presenting this tremendous amount of research, and we're looking forward to seeing what other output and results you come up with as you're moving forward with these projects.
Thank you so much for having me. It was a pleasure. I love the discussion and the questions. Everyone, feel free to contact me if you have anything else you want to discuss. I think it'd be cool to do a little more comparison of the Atlantic side versus the Gulf side, especially with gag.
Yeah, I agree. All right, thank you. Well, that concludes our seminar series for February. We're going to take a break in March, the council meetings and CDAR workshops, but look for us to return the second Tuesday of April. Thank you all. Have a good day.
Thanks, bye.